Hello friends, welcome to Bhagat online classes. Today we'll discuss the constituents of the nucleotides, okay, that are phosphate groups and the sugar molecules. Because in the last class, that is in nucleic acid part 2, we have discussed the structure of nitrogenous bases. Now it is time to go through the structure of phosphate groups, okay, and the sugar molecules involved in the structure of nucleotides. Okay, so here we have actually the sugars are of two types. Okay, the pentose sugars. Why called pentose sugar? Because the sugars have 5 carbon atoms, that is why called as the pentose sugars. And two types sugars are present in nucleic acids, that is ribose sugars, another is deoxyribose sugars. Okay, here the formula for ribose sugar is C5H10O5, whereas deoxyribose sugar has the formula C5H10O4, that is one less than the ribose sugar. Okay, so let us go through the structure. Okay, first of all we'll draw the ribose sugar. Okay, the structure of ribose sugar is like this. Here you can see the structure. Okay, we can also count the, here see C5, that is 5 carbon atoms, 1 2 3 4 5, okay, and the 10 hydrogen atoms, 1 2 3 4 5 6 7 8 9 10 hydrogen atom, and oxygen 5, 1 2 3 4 5 oxygen atoms. Clear? So this is ribose sugar. Clear? But the structure of deoxyribose sugar is just similar. It is just similar to ribose sugar. But here what happens? Only at 2 prime carbon atoms, oxygen is absent. Oxygen atom is absent. That's so the structure of deoxyribose sugar would be, here you can have just similar structure like, okay, this is the first carbon, second, third, fourth, and this one is the 5. Okay, fifth carbon atom here, hydrogen, OH. Okay, here at the 2 prime carbon atom, that is at second carbon atom, what happens? Okay, there is no oxygen atom. Clear? So here this is the structure of deoxyribose sugar. Okay, this is deoxyribose sugar. So what is the basic difference between the ribose sugar and the deoxyribose sugar? In case of deoxyribose sugar, at 2 prime position of the carbon atom or the second carbon atoms, O group is absent, okay, which is present as the hydroxyl group in ribose sugar. Okay, clear? Okay, you can draw the structure from the board here. Clear? Okay, now we'll move on to here. One thing you should know, that from these two types of sugar molecules, in case of DNA, okay, that is in the formation of nucleotides in DNA, only deoxyribose sugars are involved, whereas in case of RNA, only ribose sugars are involved. Now let's move on to phosphate groups. Okay, next is the phosphate group. Okay, okay, so next is phosphate group. Okay, actually what happens in nucleic acids, the acidic nature, okay, that is due to the presence of phosphate group. Okay, that is the phosphoric acid. You know, the phosphoric acid, the general formula is S3PO4. Okay, and the phosphoric acid is present in nucleic acid in the form of phosphate groups. How? See this. Okay, you know that phosphoric acid is H3PO4. The structure is, okay, okay, here you can have, this is phosphoric acid H3PO4. Okay, so whenever, okay, it is present in nucleic acid, it is present in the form of phosphate group. Okay, that here phosphate group is formed due to the removal of 3 hydrogen bonds, so 3 hydrogen atoms here, okay, from the phosphoric group. That is, now the phosphate group is here 3 hydrogen or 3H plus. Okay, that is by removal of 3H plus. Okay, so due to removal of 3 proton groups or 3 hydrogen, okay, ions from the phosphoric acid, we can get phosphate group, and that phosphate group is present in nucleic acid, and due to presence of this phosphate group, nucleic acid has the acidic nature. Okay, clear? So now we'll move on to, okay, so next one is the structure of nucleotides. Okay, we discussed near nitrogenous bases, phosphate groups, and the sugar molecules. Now it is time to know the structure of the nucleotides. Okay, let's move on to the structure of nucleotide. Here, okay, here we have the heading, that is structure of nucleotide. Okay, here actually what happens, you know that a nucleotide consists of 3 things. Okay, nitrogenous base, phosphate group, and the sugar molecule. Okay, so here the general structure is that if this is the here, okay, H, okay, so this is the sugar molecule. Okay, here what happens, in case of first carbon of the sugar, a nitrogenous here, okay, OH is removed, okay, and this OH is removed, and here, okay, a nitrogenous base is added. Clear? But okay, a phosphate group is like that. Okay, here OH is removed, here this OH is removed, okay, removed, and here phosphate group is added. Okay, a phosphate group is added here. Oh, okay, this is the phosphate group. Clear? Here you can see this is the sugar, okay, nitrogenous base, and this is phosphate group. Now this total structure is called as nucleotide. Okay, as you know, here at 2' or 2' position of the carbon, OH group is present. That is why this sugar is the ribose sugar, and okay, here this nucleotide is the nucleotide of RNA, that is ribonucleotide. Clear? And if there is only H atom at 2' position, second carbon position of the sugar, then now this structure, that is sugar, nitrogenous base along with phosphate group, this is a nucleotide of DNA, which is called as deoxyribonucleotide. Clear? Okay, and here you should know that the nitrogenous base, okay, the nitrogenous base, this nitrogenous base which is attached to the first prime carbon of the sugar, okay, here there is a specific connection. Here what happens, this bond, the bond between nitrogenous base and sugar is called as glycosidic bond. What is that? Glycosidic bond. And whereas the bond between sugar and phosphate group is called as ester bond. What is that? Ester bond. Clear? So this is the clear picture of the nucleotide we have. Okay, thus a nucleotide is formed from the 3 things, okay, phosphate group, nitrogenous base, and the sugar molecule. Okay, next, okay, we should know that how a polynucleotide chain is formed. Okay, we'll give a brief idea here. Okay, that polynucleotide, you can have polynucleotide. Let us consider this is the sugar. Okay, at 1' carbon is a nitrogenous base. Let us consider here adenine is attached. Okay, here at 5 carbon a phosphate group is attached. Clear? But to make a chain, okay, at 3 carbon position another sugar, okay, another phosphate group is attached, and that phosphate group is again attached to the, okay, the 5 carbon position of another sugar. Okay, now let us consider this is G, that is guanosine, this is adenosine. Okay, thus, okay, here you can have take another first prime, second prime, third prime, fourth prime. Okay, so here another sugar molecule. Okay, here you can have another, that is, see this 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. So here let us consider here T is present, thymine is present. So this is the chain, okay, of polynucleotide, nucleotide, one nucleotide, second nucleotide, third nucleotide. Thus, okay, many nucleotides join and form a polynucleotide. Here you can have, okay, here you know if this is the first, the last terminal here, there is another terminal. In this terminal here, this ends with the phosphate here ends with 5 carbon. So this is 5' end or 5' end, and here this is, okay, here, because you know that if there would be another phosphate group or the sugar, that phosphate would be attached to 3' end. So this is the end, this is the terminal, so it is considered as 3' end. Okay, so when we talk about the structure of DNA in the polynucleotide, the strands in DNA, okay, that one end is 5' and another is 3' end. Clear? So thus, okay, that is due to this condition. Okay, we can say this is the 5' end, that is the 5' end. Okay, so that's all for today. Okay, I hope you can have the idea regarding the nucleotides. Okay, and for the clear regarding the nucleic acid, you should go to our previous videos, that is nucleic acid part 1 and part 2. Okay, and to get update with new videos, new arrivals, okay, regarding any chapter, okay, you should subscribe our channel. Okay, thanks to all of you.